Hey everyone, let's go ahead and get started. Thank you so much for joining us today. I assume you can see my name, but this is Brian Helmuth. I'm Jess's PhD supervisor here at Northeastern. I want to start by thanking everybody online for joining us today and for your patience as we navigate this new normal for PhD defenses. I thought for a long time about how best to introduce Jess, and must have gone backwards and forwards about a dozen times about how to do it. Jess came to her PhD in what can probably be best called a non-traditional route. She graduated in 2007 with a BFA in philosophy and photography from NYU, and then for several years worked as a photo editor before going back for a degree in biology at UMass Boston, where she did undergrad research in Ron Etter's lab, and then graduated in 2013 summa cum laude. That timing coincided with my move to Northeastern in 2013, and she contacted me to talk about the possibility of working in my lab as a PhD student. And it really rapidly became apparent that, at least on paper, she was an excellent fit for my lab. Her letters of recommendation said things like, "This is the best student I've ever had, and you have to take her on as a student." And this is from people who don't always have a reputation of being effusive. But as any advisor on the call knows, how someone looks on paper doesn't necessarily tell you how successful they'll be as a graduate student if they're a good fit. So we, of course, scheduled an interview. And what followed was a series of bad luck. First, I got really sick, and my wife, Patrice, had to keep calling Jess to reschedule over and over again. Then I was off the field work, and then she was unavailable. So in the end, I accepted her based on the recommendation of her letter writers, taking it at face value that she'd be a good fit, never having met her before she started in my lab. And I've never regretted that decision. It was a really, really good one. Jess has been going like gangbusters ever since then. She rapidly got thrown into the fire in 2014 as part of Mission 31 with Fabien Cousteau, where our team had to spend day after nauseating day diving from a small boat to work on the Aquarius habitat. And then she was kept equally busy with the many press appearances and outreach events that were part of that program. She started her field work and, of course, didn't choose an easy topic or an easy field site, but instead decided she was going to work in the dead of winter in northern Maine. In addition to her academic work, she also really shined as a mentor to young women, teaching them everything from artistic design, which she got from her mom, to construction of electronics. And then in 2016, was awarded the College of Science Graduate Excellence in Innovation Award because of her ability to combine her research and education and outreach and mentorship in some really creative ways. She has published. She had an excellent review in 2016 in Advances in Ecological Research that's still receiving a lot of attention. Has another manuscript that we hope are in the final stages of review at Journal C Research, and has two other excellent chapters that'll also be published. Right now, on top of her grad work, she started working at a position at the Volpe Institute at the US Department of Transportation in 2018. And after graduation, we'll be moving there for a full-time position. So we're really gonna miss having her in the lab. She's been a fantastic student and a friend, and we're really glad she's staying locally. So hopefully we can continue to lure her back to doing intertidal work with her. So please join me in welcoming Jess Torossian.
Try the mic. Hello, everyone. Can everyone hear me out there?
I can.
All right. Thanks, Brian. Brian, thank you so much for that wonderful introduction. And I just want to thank everybody that's out virtually. I'm going to try really hard not to cry. I cried through my entire 8th grade graduation, and my mom was there to yell at me to stop. She's not here today in person, so I got to keep it together for myself. But I've just been really blown away over the last few days with the amount of support that I've been receiving from my family and from my friends, sort of all over the country, from elementary school all the way through college, through my PhD. It's just been really wonderful to hear from so many people. So today I'm going to be talking, as my slide title suggests, about seasonal temperature stress biotic interactions and their effect on blue mussel physiology in the Gulf of Maine. So as Brian said in my introduction, I got my start doing creative work, doing visual photography, working in the visual arts field. And I think ultimately what kind of drew me into biology and ecology is that at the start, it comes from a fundamentally visual place. We go out in the world and we notice patterns of different species. We notice patterns of where these species are distributed. And ultimately, it's up to us as ecologists to try to understand what's actually driving these patterns. What are the processes that are causing these particular species on this intertidal rock to be living in this location at this particular time in this particular place? And some of the factors that we can consider are things like abiotic or physical factors. And these include pretty common things like temperature, relative humidity, solar radiation and amount of sunlight for photosynthesizers, salinity in the water, flow, and even pH, things like how acidic or basic the water is. But for the purpose of this talk, I'm mainly going to be talking about temperature and how that's impacted, how that impacts the physiology of ectotherms. So ectotherms, unlike birds and mammals, cannot control their body temperature. And they have to depend on their body temperature from the environmental conditions that they're surrounded with. And why this is so important is because temperature pretty much underlies the basics of biological and physiological function in ectotherm physiology. It controls reaction rates. So as you see in this figure on the right-hand side, the lethal temperatures start to fall at the very extreme. So at very cold temperatures on the left, animals are going to start slowing down their physiological activity at that CT max, and they're probably going to die at that LT min and the CT min. And then at the high end of the range, proteins are going to start destabilizing. You're also going to start to see a break in physiological function at that CT max and that LT max. So this sort of captures how an organism responds to temperature. Typically, there's a peak or an optimum performance level where they're doing pretty well, and a pretty narrow band where physiological performance is happening, sort of at a happy pace. And the other reason that we care so much about temperature, especially for ectotherms, is that global climate change is causing an increase in mean temperatures generally as we've moved from the Industrial Revolution. So this is a really cool infographic. It's from Show Your Stripes, which is from Ed Hawkins. And the idea behind this is basically that each line represents a different mean annual temperature, and the degree of intensity in those lines tells you how hot or cold it is from a baseline temperature. And so as we've seen, we can see that it's increasing generally in temperature, and as temperature increases globally every year, we start to see this affecting species patterns. So many animals, like this lobster, are actually increasing their range over time, moving northward, or a poleward shift, as we tend to call it. But in some cases, we've seen many species don't quite conform to this pattern. Sometimes the shifts are a little bit more complex, they're a little patchy. Sometimes the northern range moves faster than the southern part of the range. And sometimes they're just going in the opposite direction or going downhill when they should be moving up a mountain. And there's a lot of things that can contribute to these factors, but while we're still sort of talking about abiotic conditions, one of those things has to do with how we think about temperature over large scales. And normally when we conceptualize temperature over a large geographic scale, we tend to want to draw a point, to pick 2 points at different latitudes, draw a line, and assume that there's sort of a linear gradient in how that temperature is going to change over time. But what actually happens is quite different in nature. And this is a study from Sabre et al. On the coast of Portugal. And if you look in figure B, the grand mean kind of matches that expected temperature pattern. But once you start looking at other aspects of temperature, like the daily minima in figure F or maxima in figure J, you start to see these kind of really weird mosaics that are not linear, and even sites that are really close together on the coast and have very similar latitudes may actually be experiencing really different conditions. And as you drill down into these sites and you kind of get even further, you start to see how even over a scale of millimeters or centimeters, there's going to be changes in temperature. So if you're one of those ectothermic animals like that sea star that we had in the previous picture, you could live on the top of the rock where it's really hot, but that might not be such a good idea. So you may want to kind of take advantage of the thermal refugia that form in some of those cooler blue temperatures. Try to hide yourself from the conditions, because otherwise you're probably just going to bake in the sun and die. So in addition to these abiotic conditions that can vary at different spatial scales, there's also biotic factors that can vary at different spatial scales. So moving from abiotic factors to biotic factors, there's a variety of different ways that other species can shape species distributions. And so this includes things like competition between maybe a species of alga and the barnacles on the rock, as well as consumption, either from primary consumers like these snails on algal films or a sea star on a barnacle. And typically we think of these abiotic factors that control a species' physiological tolerance and the biotic factors that affect patterns of local abundance as being kind of separate, and that they overlay like a Venn diagram. But in reality, they actually strongly interact with each other. And so you can see that sort of the way that these two aspects of a species niche interact is through energetic feedbacks. And this is an example from Wooden et al. 2013, where the figures on the left are showing a model that was run using only temperature. And you can see how it does a pretty good job predicting where blue mussels are going to be on the east coast of the US, but it doesn't do a very good job when it gets moved to Europe, and that's in the bottom left figure. And part of the reason is that it's not really considering the food supply for these mussels. So in the right-hand side of the figure, that's actually an energetics model. It's taking into account how these mussels are utilizing food and how much food is available for them in the environment. And it actually does a much better job of predicting how these species are going to be distributed upon the European coast. And as we move into predicting how species are going to respond under climate change, this becomes even more important. This is an example from Briscoe et al. That used an energetics model and compared it to a correlative model that's just looking at temperature and other environmental conditions. The colors indicate how similar or dissimilar those two model predictions are, using either means, which is in figures A and D, or extremes, which is in figures B and E. And you can see that depending on sort of where it's trying to make these pattern predictions, the energetics model and the non-energetics model can differ significantly. So overall, we've just gone through my introduction and pulled some concepts in from my fourth chapter. Next, I'm going to be talking about my chapter 3, which Brian mentioned is in review. We're not going to be talking about. And that leaves us with chapter 1 and chapter 2 to discuss today. So both of those chapters happened in the Gulf of Maine. So I just wanted to orient you. Most of us are here on the east coast of the US right now. The Gulf of Maine runs from the Bay of Fundy in Nova Scotia at the top right of this figure, all the way down into Cape Cod Bay at the bottom left. And then when we're looking at that sort of global trends figure as well, comparing the global trend to what's been happening on land in Maine, you start to see that in general, Maine is also warming, but there's a little bit more variability, as I mentioned. As you start to go from large scale averages down into regions, things start to get more and more variable. And further, the actual Gulf of Maine itself, the body of water, is warming faster than pretty much any body of water on Earth right now. And this is just showing the global sea surface anomaly that's been occurring since 2004 in the Gulf of Maine. So my study species that I decided to look at for all 3 of my experimental chapters is the blue mussel Middle edgeless. These mussels form really dense beds and provide a lot of habitat and are very commercially important. And if you've been talking to any researchers that have been working in the Gulf of Maine over the last 30 years, they're going to tell you that mussels in the Gulf of Maine have actually been declining in populations. historic sites that have been sampled for years, where there used to be tons of mussels, are no longer there anymore. And so this figure in the bottom right is from a paper by Sort et al. In 2016. And you can see the mussel abundance in those gray bars from the early 1970s is much, much higher than the mussel abundance in the black bars. That's from the mid-2010s. But one of the things that this study sort of ignored was that there's still a lot of mussel beds in the Gulf of Maine in soft sediment habitats. And that SORT study primarily looked at mussel beds on sort of open coast, rocky shores with high wave energy. But throughout the Gulf, there are still these very highly abundant beds that exist in soft sediment. In these habitats, the mussels sort of attach to each other or to small cobbles because there's not a lot of other sediment for them to attach to. But even in this bed in the northern Gulf of Maine, you can actually see that even though there's rocks present, the mussels are absent. And the way that these beds look between the north part of the Gulf and the southern part of the Gulf is quite different. The mussels in the southern part are dealing with much more urbanized areas. Up these embayments, there were active waterways. In the bottom right picture, that's Portsmouth Harbor, that's the Piscataqua. The naval shipyard is in the background. These mussels have to deal with large tankers delivering oil and salt every day. Whereas up in Maine, other than lobstering traffic and some fishing vehicles, they tend to be a little bit less perturbed by human influence. And there's a bunch of different pros and cons sort of for why these mussels may want to live in these soft sediment beds. And there's a whole host of them ranging from the abiotic and the biotic. But what I'm going to do today is focus mostly on the potential of the mud to act as a thermal buffer for both heat stress in the summer and cold stress in the winter. And so this brings us to my first chapter, which is trying to understand how substrate, so mud or rock, mediates temperature effects and affects regional distributions of Middleus edulis in the Gulf of Maine. And to do this, I'm taking sort of a three-pronged approach. This is sort of adapted from the climate adaptation literature for humans, where I'm looking at exposure, sensitivity, and vulnerability across multiple populations. So I'm going to start by talking about exposure. We're going to be in the first chapter for a while, so I'm going to try to keep reorienting you as much as I possibly can. So when we're looking at exposure, that's just trying to define the seasonal and regional patterns in thermal stress, both on muddy surfaces and rocky surfaces in these soft sediment beds. And for this study, I picked 3 sites in the northeast region of the Gulf of Maine, and 3 sites in the southeast region, or southwest region, sorry, I keep doing that, Gulf of Maine, with the goal of having sort of a fully factorial replicated design. But ultimately, with some temperature logger loss, it didn't quite end up being as perfect as this diagram suggests. But to measure temperature, one of the things that's sort of a hallmark of the Helmuth lab and is becoming more common in research groups is to try to modify the temperature loggers that you're using to reflect the body shape, the size, the sort of the physical thermal properties of the animal that you're studying. And so in this case, I made robomussels, which basically uses a digital temperature probe that's embedded in a mussel shell, covered in epoxy, and then deployed out into the field. The other pictures kind of show the nuts and bolts and the guts of these temperature loggers that I designed. But by doing this myself, I saved, probably $4,000 as opposed to buying the temperature loggers from the source that we normally would. To deploy them, I put them in 6 blocks at each site. The mud loggers were attached using a zip tie to a garden stake, so they sort of stayed at the surface of the mud. And the rock loggers were attached using Splash Zone Z-Spar marine epoxy. So next, I'm going to walk through how I looked at patterns of thermal sensitivity across a subset of these populations. And I did this by looking at lethal temperatures at both the cold end in the winter and the hot end in the summer. So this study used 5 of the sites for the summer experiments and 4 of the sites for the winter experiments. I also crossed site with exposure. So I wanted to understand how repeated exposure to stressful temperatures would impact these lethal temperatures for each population. And so I created 6 replicate curves per site, which I'll explain a little bit more what that means on the next page. And I calculated them once in the winter and once in the summer. So the upper lethal temperatures that I was looking at, we use 5 temperatures. We create these sort of lethal temperature curves. So each replica involves 5 mussels per temperature that we look at. Then we fit what we call a logistic regression. That's just this swoopy curve on the right. And then using a function in the R statistical language, we're able to estimate the temperature that is predicted to kill 50% of the population from our experiments. For these LT50 curves, I exposed the mussels for 2 hours for each exposure, and the exposures were 24 hours apart. Some of the values that we got in our experiment are a little bit higher or lower than what we've seen in other experiments, looking at thermal sensitivity in mussels. Part of that is because the exposure time that we used is a little bit shorter, just to represent the ecological relevance to the mud beds that we were sampling from. And so all I really want to talk about in this initial figure for LT50 is sort of this overall pattern that you see both in the winter and the summer and across all the sites, that as you increase exposure to stressful temperatures, you start to see an increased sensitivity to temperature stress. So mussels are 50% of the population is predicted to die at a warmer, less negative temperature in the winter as you repeat exposures, and 50% is predicted to die at colder, less extreme heat temperatures in the summertime. So then taking the exposure results, which I will cover next, and those sensitivity values that we just calculated, I wanted to look at vulnerability of the mussel populations. So to do that, one step was to sort of compare those lethal temperatures to the patterns that we were seeing in the field. And so just to reorient you again to this figure, I'm going to be talking about the winter set first. Anything with a little snowflake in the right-hand corner is going to relate to my winter experiments. The southwestern sites are in that orange box, and the northeast sites are in the teal box. So when you look at winter temperatures, other than TF, which is sort of anomalously not very stressed out by cold temperatures, there's not a lot of differences between sites, other than sort of that continually decreasing sensitivity. But TF does differ significantly from the other populations in terms of its lethal thresholds. Then when we look at the exposures that we saw in the field, for this figure, I'm representing sort of 2 years of data across both regions. So the top figure is from winter 2015-2016. The bottom figure is from winter 2016-2017. And if you look on the right-hand side, that's sort of your legend for the different sites. So mud has the open points, rock are going to be the closed points, and then color and shape are going to tell you which site that they're from. So you can see just sort of across both regions, and especially during those sort of extreme cold snaps, mussel loggers that were in the mud tended to experience elevated temperatures compared to the rocky counterparts. Further, when we overlay those lethal temperatures that we calculated, we start to see how this could lead to some significant mortality events at these particular locations, but that it's not happening equally in all regions at all times, and it's also not happening across all substrates equally at all times. So first, looking at the rocky loggers in the southwest during this particular time, we would expect to see a significant mortality event only on rock in these locations. And then when you look at mud, there's only a couple times where mud loggers would predict a significant mortality event. That's in February of 2016 at OP, which is one of the southern sites, and also in December of 2016 at WR and also possibly TF, which are both northern sites. And one of the main hypotheses behind this discrepancy between mud temperatures and rock temperatures in the winter has to do with the way that the ice pack forms in these habitats. So you can see in the top right video that just played, sometimes the ice is so robust that you can actually do penguin slides across it. There's actually a whole mussel bed underneath there. Even within a site, as you see in the bottom left figure, there's really patchy ice cover. So you might not actually, if you're a mussel that happens to live in one of those bare patches, you may actually be less protected. And then in the two right-hand figures, just sort of showing the difference in strength of that ice cover. So in the top figure, I could just reach in and grab that mussel. It's unclear how well that ice is buffering temperature. Whereas in the bottom figure, for some reason, I'm barehanded, but I also needed a hammer to break through the ice pack. And you can sort of see the temperature data looking at daily minima support this hypothesis as well. Sort of earlier in the season, you tend to see more similar temperatures between both substrates, but as the winter gets colder and that ice pack potentially starts to form, you see more extreme temperatures on the rock, which is at that red line, than you do in the mud, the blue line. So now switching over to summer, which I think most of us are probably ready for at this time in the spring, we're going to look at the lethal hot temperatures. So that's again, northeast is in teal, southwest is in orange. And you can see that when you just expose these mussels to these stressful hot temperatures just once, there's not a lot of difference between the sites. And that's what you see in those black bars. But as you start to increase exposure a second or third time, differences between the sites do emerge. And they don't seem to be based solely sort of on latitude. For instance, the three southern sites have very similar tolerances, but there's actually a significant difference in tolerance between the two northern sites. When we look at summer, the temperature effects don't seem as dramatic. The model showing that there is a difference between site and there is a difference between substrate, but it's much, much more minor than what you would see in the wintertime. So overall, the pattern is a little bit less predictable. And that just might have to be due to the fact that solar radiation plays more of a factor in summer temperatures and how the sites are facing, or maybe composition of the substrate might affect those temperatures that we're seeing. When you add in the lethal temperatures, unlike winter, you actually don't see any of the sites crossing that lethal threshold. So during the periods that we surveyed, it actually seems like winter temperatures are potentially much more stressful and much more vulnerable time for these mussels at these sites. To sort of try to validate some of this in the field, I wanted to also do a caging study. This is to further look at vulnerability. And to do this, I picked 4 of my sites, 2 in the northeast, 2 in the southwest. And I wanted to look at how substrate and season, as well as caging, and I'll talk a little bit more about caging as well, influenced the patterns of mortality that we saw. And so this was done with the intention, again, of having 6 blocks per site for a total of 36 plots per site per season. And so to do this, we moved live mussels around. We tried to clear the mud areas as best as we could. We scraped the rocks off. As I mentioned before, at some of these locations, the mussels are not on the rock, so we had to do a transplant experiment. We allowed them to sort of attach under intact cages for 24 to 48 hours before removing parts of the cage to make these cage controls. And you can see in the bottom figure that they actually were able to attach on their own. This is sort of the results of one of the experiments. And the reason that we use the cages for this experiment is that we wanted to try to get at whether temperature was driving mortality at these sites or whether some of the other factors, another physical factor, ice scour in the winter has been shown to scrape the mussels off rocks, and predation stress, especially in the spring, summer, and fall, can be significant. During the setup for our experiment, when we only had rock cages out across all the sites, we actually experienced a really early cold snap that actually ended up killing most of the mussels at these locations. So we had to sort of set things up again. But when we're just looking at the results from the rock during the setup experiment, we do see significant mortality across all sites in the region, though the mortality at PI, one of the southern sites, is a little bit less than we see in the other regions. And when you look at the temperatures on the right-hand side, especially at those northern sites, TF and WR, which are in green and orange, green and yellow, sorry, you can see that both substrates are predicted to be, below -15C, which is going to cause mortality above 50%, which is what we see in this study. However, unfortunately, when we ran the main caging experiment, we only saw differences. Let me start that again. When we ran the main caging experiment, the main thing that we saw was that the temperatures were not very extreme. In fact, during the main run of the winter experiment as well as the summer experiment, we had very kind of mild weather. So we weren't expecting to see very high mortality at all. And that is what the data shows. There's not a lot of mortality. However, when you run the model, we do start to see differences between sites. So for example, OP, the southernmost site in the study, had higher mortality, lower survival overall, and WR actually had the highest survival, and that's the northernmost site. We also saw an interaction between caging, site, and season. And this is sort of a little bit more complicated to untangle. In the winter, we see lower survival in the northeast in those rock controls. My hypothesis is that it's that same ice that's being really good for buffering thermal stress in the mud is probably also acting as the tide comes in and out to pull those mussels off the rocks. And so the mussels might be surviving, but it's actually physically removing them from that surface. And then in the summer, in the southwest, we actually see a significant effect on both rock and mud controls. This could be potentially due to higher predation rates in the south, which many studies have documented, but it could also be due to sort of repeated exposure to sublethal stress. And we did have some temperature loggers in those cages, so looking more at kind of what might be driving that is something that we need to do. So we did it. We got through chapter 1. So what did we find? When we looked at sensitivity, we saw that there were interpopulation differences in lethal limits, and that those are only apparent after repeated exposure. When we looked at exposure alone, we saw that mussels on muddy substrates tended to experience less extreme temperatures at both the high and low end of the range. Well, this was more apparent in the winter. And we also saw that there was a lot of variability kind of in the timing of how those extreme temperatures hit. And when we put those two things together, we saw that with vulnerability, it means that substrate-mediated differences in thermal stress can impact mussel survival differently, both within populations in terms of substrate, but also between populations, just because there's slight differences in sensitivity and also the thermal patterns that they're experiencing. But based on that final caging experiment, we saw that we still need a better understanding of how repeated sublethal stress and other abiotic and biotic factors could potentially impact survival. And so that leads me to my second chapter, which is trying to understand how freeze-thaw cycles impact sublethal short-term energetics in mussels. And to do this, I just picked one site. So we were just looking at WR, which was that site that we saw in yellow in all the other figures. One of the reasons that I wanted to look at these sort of freeze-thaw cycles is that when we think about climate change and we think back to that figure that I showed at the beginning of the talk, we typically think about it as being a linear pattern where we're increasing in heat. But it's actually affecting winter conditions as well. And the way that it's doing that is sort of unpredictable. Sure, you're seeing increased mean temperatures, but what I'm focused on in this talk is how it might impact air temperature variability as well. We might see more intense cold snaps that happen earlier in the year. We might see some later cold snaps that might be unseasonable, and we also might see unseasonal warming. And when you look at the terrestrial literature, there's a lot of studies on this. There's a lot of studies on plants and how they respond to frost and freezing. There's a lot of studies on insects that are overwintering in terrestrial habitats and how they respond to snowpack and sort of warm periods in the middle of winter. But I think partly because the marine environment seems relatively immune to those terrestrial winter conditions, the intertidal kind of gets skipped, even though mussels that live in the intertidal are covered by water a significant portion, but they're also part of the terrestrial world for part of their life. And so for intertidal species, we don't even have many baseline studies that even understand how daily or maybe every other month or bi-monthly exposure to air conditions impact how they behave when they're resubmerged at high tide. And so to do this, I collected mussels from WR in northern Maine. And I wanted to look at a few things in particular. I wanted to look at water temperature, because as we saw from that figure I showed earlier, the Gulf of Maine is generally warming, and this is sticking around throughout most of the year. I wanted to look at aerial exposure temperature. And I also wanted to see how this changed over time. So day was also a factor. I had 2 levels of water temperature, a relatively normal temperature for the late spring around the time I collected, though up the embayments I was looking at, it might be slightly colder. So 5.5 C and 8.5 C. And then I also looked at how a sublethal cold exposure, so minus 2.5 degrees C, so a negative temperature exposure would impact a few different metrics. And so I did this on 2 days. So this is a rough schematic of how this worked, though they were only exposed again for 2 hours to try to keep the exposure time sort of ecologically relevant. Controls were exposed at the same water temperature as air temperature. The cold exposures experienced daily exposures to sublethal stress, and then at the end of the experiment on day 8, the mussels that were previously exposed to cold temperatures were exposed to a warming cycle. And on both day 1 upon resubmergence and on day 8 on resubmergence, I measured 3 metrics. I looked at respiration rate, which is just oxygen consumption in the water. It's similar to me measuring respiration rate on any of you guys. This helps get at overall energy demand for maintaining the animal. I looked at clearance rate, which sort of calculates how well mussels are extracting food from the water. So mussels are filter feeders, and they feed on algae in the water column. So clearance rate sort of says how fast they're clearing that water and removing those particles. And then I also looked at assimilation efficiency, which basically just measures how well they're actually able to take in the energy that they're potentially removing from the water column. So they don't necessarily digest well everything they eat, sort of like us. And so we can look at how that energy is acquired at under different conditions. So the results for this, I'm going to show you 3 graphs, one for respiration, one for clearance rate, and one for assimilation efficiency. For respiration, what we saw was that at day 1, aerial temperature significantly impacted respiration rate, but we didn't see an effect of water temperature. And for this particular metric, I had to do 2 different models, one on the first day and one on the second day. So it's going to look a little bit different than the other figures. On the y-axis in this figure is oxygen consumption, and on the x-axis is water temperature. So you'll see that oxygen consumption is increased in mussels that were exposed to that cold minus 2.5 degrees C air exposure relative to the mussels in the controls, at both water temperatures. But when you go to day 8 and you do this again and you add in that warming cycle, the effective aerial exposure is no longer impacting respiration. And there's a few different potential hypotheses for why this is occurring. Typically, when mussels are exposed to air in hot temperatures, they will often elevate their respiration when they're resubmerged, usually because they're enacting anaerobic metabolism. There's been some studies that show that molecules that help maintain protein conformation, so help keep the mussels alive, are produced in other species of mussels at cold temperatures. And also mussels will sometimes breathe from some of the liquid that they have trapped in their body when they close up. And so if that's slowly starting to freeze, and especially at those cold temperatures, that oxygen may not be available for respiration. The next figure is looking at clearance rate. So in this figure, since water temperature was not significant, you're only seeing since air temperature was not significant, you're only seeing water temperature. So The gray circles represent the low water temperature, the black circles represent the elevated water temperature, and clearance rate is on the y-axis. The x-axis is showing you experiment day. And so what you can see is that water temperature significantly affects respiration rate. And even though it does look like experiment day could be potentially significant, that was not confirmed by the model. But in general, you are seeing a slight increase in rates as we move throughout the experiment. So in this case, clearance rate in mussels held at the higher water temperature was elevated upon resubmersion when compared to colder water mussels. And then lastly, we looked at assimilation efficiency, which was measured using a study that was developed by Conover in the 60s. And this basically was sort of revolutionary. It allows you to look at how well mussels are assimilating their food that they're taking in without having to capture every bit of feces and account for every single particle eaten. So it uses just sort of the ratio of the organic content, that's that AFDW, that means ash-free dry weight. So that's the carbon content of the food compared to the food with the full organic content in it. And you do the same thing for their feces. So I had to take little filters and collect a lot of mussel poop to be able to do this part of the experiment. And what's interesting is when you look at assimilation efficiency, the two effects that we saw for clearance rate and respiration rate are no longer a factor. So aerial temperature doesn't affect assimilation efficiency, neither does water temperature, but experiment day does significantly affect how well these mussels are assimilating the energy that they're obtaining from the food. And you do see a slightly higher, very slight higher increase in assimilation efficiency in colder water mussels at this time. And so looking at chapter 2, what we see and what I saw at the time was that you see different responses depending on the physiological metric that you're looking at. So when these mussels are undergoing these daily freeze-thaw cycles, aerial temperature could potentially be impacting respiration rate. Water temperature was impacting clearance rates. And then how long they've been going through this exposure regime can also impact how well they assimilate that energy. And what this means for the field is that these mussels could potentially be acclimatizing to those intertidal conditions. If they're sitting at the mean tide line, which is where I collected some of these mussels from, they're not going to be exposed to air every day of the month, only during some of those more extreme tide periods. So overall, this chapter sort of indicates that sublethal stress can be very stressful. It can increase energy demand. And then if we think about it in conjunction with food availability, we need to consider the sort of cumulative stress that might be impacting this individual over the course of a winter season that could potentially impact growth and reproduction in the spring. And so with that, taking those two chapters sort of together, we're going to sort of think back through what we've already covered and talk about the overall conclusions between the two chapters, which is that the differences in interpopulation exposure to stressful conditions, as well as interpopulation differences in sensitivity, can play a significant role in sort of how these populations are structured. Sensitivity to abiotic and biotic stress can interact with these patterns of exposure and drastically affect how vulnerable these species are across their range and also within a site within their range. We also saw in chapter 2 that sublethal stress to aerial conditions in the winter, as well as winter water temperatures, can sort of impact the overall energetics, how much they're feeding, how much energy they need to make up for some of their aerial exposure stress that they were undergoing. And so what these two things taken together, as well as some of the lessons from my 4th chapter, and even things I'm seeing in my 3rd chapter, which I didn't talk to you about, just sort of make us more aware that we need to carefully consider the patterns of exposure and sensitivity at a variety of different spatial scales and try to understand how these interact when we're modeling species distributions and trying to predict their responses to climate change. And so I have made it through my virtual defense, which would not have been possible without like a whole host of people. So thank you so much for making the last 7 years like really amazing. I owe a lot to a crazy support system of family and friends, the Helmuth Lab. I have a lot of people on this acknowledgement slide. And I feel like someone accepting an Oscar. If I forgot anyone, I'm really sorry. There's like more people that have contributed to my success than I can even count. And yeah, last but not least, my parents, aka Dad Turn, the best intern in the world, and my Beach Plus buff family, my husband, Kirby, and then the rest of the PhD pack that's been living with me. So thank you, and I will take questions.
Yay, I'll clap for everybody.
So how it's gonna, oh, this is my voice. Hi, I'm Francis. This is how it's gonna happen. You guys are gonna type in questions. I'm gonna read them out loud, and Jess is gonna answer them. We have a lot of hooray. Oh, here. Hooray. Go, Jess, go. Oh, we do have a question.
Okay.
What does interpopulation differences in sensitivity mean?
So so interpopulation differences in sensitivity can mean a few different things. For the purposes of my study, I wanted to.
Hey, Jess, there's a part 2 to that question.
Okay, what's the second part?
The second part is, does that mean differences in genetics?
That's what I was just going to talk about.
Okay, sorry.
So yeah, so it can mean different things. For the purposes of this particular study, I wanted to understand sort of that immediate ecologically relevant response. And so I purposefully didn't try to even untangle the genetic portion of what was going on here. I actually tried to prevent acclimation in the lab as much as possible, because I tried to have the mussels basically just come right out of the field and go right into these sensitivity experiments. Because in the field, there's a bunch of different things that can lead to population differences. As the person asking the question suggested, one of those things is genetic differences. This is typically when animals are seeing localized adaptation where populations are under different stressors, and the success of different individuals is causing that genetic makeup of those populations to change over time. We don't think that that's much of an effect in the Gulf of Maine for this specific species, just because for Middleus edulis, they're a broadcast spawning species. So what that means is that they release sperm and egg into the water column. It fertilizes in the water column, and then their planktonic larvae kind of drift in between sites. So it seems unlikely that there's genetic differences, just because other studies have shown that the populations in the Gulf of Maine are relatively genetically homogeneous. However, there are also some studies that show that a neighboring species, Middleus trossilus, can hybridize with Middleus edulis. And so there's actually a gradient of trossilus genes as you kind of move from Canada down the coast of Maine. So it's possible that there's some trossilus intrusion creating more robust responses to cold stress. That's not something that I specifically was able to look at. It's also possible one of a UMaine graduate student just released a dissertation a couple of years ago looking at how the flow patterns, a very intricate flow diagram along the coast of the northern part of Maine, affects propagule movement between these mussel beds. And there actually looks like there's more local retention potentially of larvae than we thought. And so that could also lead to maybe genetic effects, but if not genetic, maybe some familial effects or kind of intergenerational differences. So there's a lot to still look at in untangling what's driving those interpopulation differences in sensitivity.
Okay, that was a question from Elizabeth Moran. This is a question from Mark Patterson. Oh, wait, let's hold on. Elizabeth Moran has a part 2 question. So,
Elizabeth is my college roommate, for those of you playing at home.
How do you tell if a mussel is dead or alive?
So for this study, I basically looked at a few different metrics to determine mortality. You can poke the tissue or the foot, and if it moves, that'll tell you if it's alive or not. For some of these, they were starting to get so stressed out that I was actually allowing them an hour to kind of respond before determining if they were dead. So in a couple cases, I would poke them, and it would maybe take them up to 40 minutes to sort of reclose. So before I re-exposed them, I really wanted to make sure that they were dead. Something else I didn't mention in this talk was that I started documenting whether or not in between exposures, the mussels were producing new byssus threads, which is how they attach to the rocks, or if they were producing feces. And typically, I could tell when most of a group at an exposure was going to start dying is because they stopped producing feces and they stopped producing byssus threads, which meant that they were probably energetically stressed.
Here's a question from Mark Patterson. So to restore coral populations, Ruth Gates was working on selective breeding to reseed corals that were temperature tolerant back to the reefs. Is there a concern among aquaculture folks that selective breeding or replanting from a good hardy source populations is needed to keep rope aquaculture going? How might you advise them to do so?
I think that's a really complicated question, Mark, as I'm sure you intended it to be. I haven't talked to the aquaculture folks that so the people at the Down East Institute, I've spent a lot of time there. They do some mussel breeding. That's not something that we specifically talked about. Phil Yund, who's on my committee, was also doing some work trying to see how they could interbreed mussels from different populations and what that did to the genetic compositions in sort of lab experiments. I think that there's the potential that you could try to do selective breeding. Even if you did come up with a species that was potentially more or less resilient, or sort of a sub part of the species that was more or less resilient to certain conditions, you'd also have to be concerned about what the conditions are that they're experiencing in the field and sort of how those genetics interacted with just sort of the pattern of exposure. So another thing that is a minor caveat or sort of a major caveat to the sensitivity analyses that I did in the populations is that we can see in the field, we call it acclimatization, depending on the season and how those seasonal temperatures start to move in, mussels can become more or less stressed, and this happens across a variety of species of ectotherms. They can become more or less tolerant to those conditions depending on how long they've been exposed to them. So you would need to make sure that whatever sort of genetic population you were trying to breed was actively going to be experiencing those in the field. And then you also would want to make sure that their propagules, that their larvae were actually going to be seeding beds that also had potentially similar conditions. Otherwise, you'd just be sending maladapted larvae to new sites. So there's sort of a lot that could be going on there.
I have a question on YouTube.
Okay.
Sarah Donlan asked, I'm thinking about the tidal amplitude in Massachusetts versus Maine. Do you think tidal amplitude could affect mussel sensitivity to cold temperatures? Were these mussels from similar tidal heights?
Yes, so the mussels were from similar tidal heights, although in Maine, you do actually see some of these populations occupying slightly higher parts of the intertidal, just because in the summertime, although our temperature loggers didn't actually show it, I actually expected the summer temperatures to be much less extreme in northern Maine because there's often this heavy fog that rolls in. And it can really sort of deflect solar radiation. So I tried to collect mussels from around Maine at all the sites and deploy loggers at similar levels. Thermal sensitivity and exposure is a function of how long the tide is out, how long are they exposed to those air conditions, and sort of what the wave splash and humidity conditions are as it starts to roll back in. So tide height is definitely a factor. But yes, mussels were tried to be collected from around the same tide height. And at these soft sediment beds, they tend to occupy a similar space. It's a little bit different than what you see on sort of wave exposed rocky shores.
I have a question.
Okay.
So when you're in the soft sediment sites, I noticed there were rocks around too as well. And I noticed that most of the mussels were congregated on the mud, not on the rocks. How do you feel like the data you're showing with the sensitivity kind of relate to what we see?
I think, yeah, so I think what I showed with vulnerability, looking at sensitivity as well as exposure is part of that story. And I think what that caging study at the end is also trying to get at also is part of that story. So it seems like there's sort of two physical effects happening on those rocky surfaces at these sites. One is temperature, that is getting way colder in the winter and causing much more likely to cause mortality events. And the other part is that ice scour that I talked about, where there's the potential for sort of physical scraping of the muscles to fall off. And the other thing that is important to consider as well is that those byssus threads that I talked about is how they attach to rocks. There's a study by Emily Carrington from New England from a few years back, I want to say like 10 or 15 years ago, looking at how winter conditions are actually not very conducive to the production of byssus threads. And so mussels tend to see more drop off in winter conditions as well. So that loss from those substrates in the mud might be due to a variety of factors.
There's a comment on YouTube by Coronavirus. It says, good luck. All right, here's a question from Nick Kovard. Nice job, Jess. In hindsight, what would you change, if anything, in your experimental design if you were to redo chapter 1 or 2?
Oh, I would change so much, Nick. The easy answer, and this is something I've talked about with so many people, is that I would have an army of interns, and an army of interns would allow me to sample like everything simultaneously. I think the caging study, I would really want to retool a little bit. I would maybe try to sample over a broader area so that I could actually deploy all 3 types of cages, a full cage control and an open plot. Since I've been doing work in the transportation sphere, I've been talking to a lot of people that work with freshwater mussels. And one of the things that they use for freshwater mussels, because they're an endangered species and they have to be able to account for the mussels that they transplant, is that they use those pit tags. But instead of like the dinky little one that we have in our life. So pit tags, for those of you who don't know, are like if you have a pet, like a cat or a dog, they're those little like grains of rice shaped things that are a radio transmitter, and you can like inject them into your dog. But you can also put them out in the field and they last for a really long time. We just never had really good luck with them, so I didn't think to use them in our lab. But talking to people who work for consulting companies that are certified to handle endangered freshwater mussel species, what they do is they use dental cement to adhere the pit tags to the surface of the mussel. Those pit tags are basically not going anywhere. And then they sort of amplify the signal from that radio receiver. And they can actually just go out with something that looks similar to a metal detector and kind of wave it over their field site and find those mussels. And so if I were to go back and do it right now, I think I would honestly do that to my caging study. I would tag every single mussel, or as many mussels as I could. And that would allow me to actually account for whether or not they just dropped off and survived, or if they were picked up by ice and maybe carried, 10 or 20 meters down the bed. It would help account for a lot of that. Because one thing we saw in these sites is, especially because they're so low wave energy, if mussel loggers popped off a rock or we lost temperature loggers, they'd often stay relatively close by. And so I think that those pit tags would be a real game changer.
Here's a question from Ron. Are LT50s different from mussels of different size? If smaller mussels are more sensitive to your temperature, this might prevent them from recruiting and help explain why they're not on rocky substrate in Maine.
There's sort of conflicting evidence in the literature about how size affects lethal tolerance. One thing that I still need to do with this data is that as the mussels died in my experiments, I recorded their body temp. I could record their body size. And so even though all the mussels were from a relatively similar size range, I did actually record which ones died first and how big they were. So one thing I could do potentially is reanalyze the data to see if there's a correlation between mussels that tend to die early and their body size. Especially at cold temperatures, you would think that the larger volume of salt water that they're able to trap, that would potentially slow the freezing rate of that water. So that's a potential explanation. Size definitely does come into play into how body temperature changes. Our mussel loggers were designed for the size range that I saw most prominently at these sites, but certainly younger mussels can be more sensitive. Their temperatures are gonna come to equilibrium and get higher faster than a larger mussel.
You answered Brian's question, so that's good. Any other questions, anyone? We have some yay, Jess, awesome Jess. Oh, new question. Oh, great job. Well done, hooray. clapping.
Okay.
Okay. So, here's another question. Elizabeth Moran asked, which was your favorite site?
I feel like the other questions were from Ori, and this one might actually be from Liz, but I don't know if that's true or not. Which is my favorite site? It's like trying to pick a favorite child. I think honestly, WR might have been my favorite site, West River in Addison, Maine. It was a pain in the neck to get to. It has like a ton of mud, and I used to get like trapped in it all the time. We lost many an intern from the Down East Institute there in the fog, but it was just so pretty and so quiet and so peaceful. But Tidal Falls comes in a close second. It's got like cool tidal rapids, and there's a lot of food there. So sometimes there would be like feeding frenzies with fish and seals and stuff. So that was also a really cool location.
With that, we're going to wrap up. We're going to clap for Jess. If the committee, we're going to take like a 5 to 10 minute break.
Let's go till about 4:10. All right.